Hello, everybody. We are going to talk about endocrine signaling today, and we're going to talk about feedback, and we're going to talk about signaling in plants. So taking a look first at endocrine signaling. Signaling molecules send out messages. They can be external to the organism or they can be internal to the organism. When we're looking at pheromones, pheromones are going to be molecules that are sent out into the environment. And that's usually done for a lot of different reasons. It could be to mark trails to the food. So in the summertime, you might see ants kind of creating a trail that leads to the food. And so they're going to, the ant that finds the food the first is going to kind of leave a pheromone trail that lets all of the rest of the ants find the food. So you might notice a long trail of ants all walking in single file. That's because one ant has put down a pheromone trail and the rest of the ants are kind of following along and picking up that scent. It could also be to define territories. So animals might mark their territory by peeing on things. So if you've got a dog that has to pee on every single mailbox in the neighborhood, he's kind of marking his territory. It can also be used to warn off predators, to attract potential mates. So kind of sometimes people are attracted to people and they don't know why. They could just be responding to the pheromones that they're giving off. So pheromones are chemical signals given off to the environment that allows other organisms to pick up that scent and respond to that signal. When we look at the endocrine system is sending out messages that go throughout the body, and it's used to regulate our body systems. There are a lot of different tissues and organs involved in the endocrine system, and they're spread kind of throughout the body, and so it's not like one specific location, but there's lots of hormones that get sent out through the endocrine system. So we're going to talk about a few of them today as examples of feedback mechanisms. So endocrine signaling is going to be long distance signaling, where you're looking at hormones being secreted into the bloodstream, and then they get circulated throughout the entire body. And only the cells that have receptor sites for that particular hormone are going to pick up and receive the signal and respond to that signal. Endocrine hormones are usually working to maintain homeostasis or to regulate growth and development. So it works a lot in feedback mechanisms, negative feedback mechanisms to maintain homeostasis. If we look at this diagram here, you've got endocrine signaling, where you've got hormones and signaling molecules being released into the bloodstream. You've got paracrine signaling, where the molecules are going to be more local regulators and they're going out to cells in the nearby vicinity. Autocrine signaling, you've got the cells that are releasing the signals are also picking up the signals, and then that's causing a response. And then we've got synaptic signaling and neuroendocrine signaling, and that's involving neurons where they're sending signals. So a synaptic signaling is going to be a chemical message going across a small space in the synapse, and we're going to talk more about that a little bit later when we get to talking about the nervous system. So again, paracrine and autocrine signaling, we're looking at local regulators because those are going very short distances. And then autocrine signaling also going very short distances, so local regulators. And so this first diagram is showing you the endocrine signaling, long distance signaling, and the paracrine and autocrine signaling, short distance signaling. And synaptic signaling also going to be short distance signaling because those neurotransmitters are going from one neuron to another neuron, and they're traveling chemical messages across that small space between them. So those chemical messages that are going through the synapse, those are called neurotransmitters. So neurotransmitters are the chemical messages that connect one neuron to another. And again, we'll talk about that more when we get to talking about signaling in neurons. Okay. One important thing when we're looking at hormones is understanding the difference between lipid-soluble hormones and water-soluble hormones. Lipid-soluble hormones are going to be your steroid hormones, and so they can, because they are nonpolar, can pass through the cell membrane. So the receptor site for those are going to be on the inside of the cell because those molecules can pass through the cell membrane. Water-soluble hormones cannot pass through the cell membrane, and so their receptor proteins are going to be on the surface of the cell membrane. So those are going to be proteins, polypeptides, amines that can't pass through the cell membrane. So those are going to have the receptors on the surface. So here you can see those would be your water-soluble hormones in that first diagram. And then here you've got your lipid-soluble hormones because they can pass through the cell membrane. The receptor protein is on the inside of the cell. If we look at the hormone epinephrine, also known as adrenaline, that's your fight or flight hormone. And that's going to be an example of a hormone that is going to be released into the bloodstream, and it's going to cause effects in the body. And epinephrine is going to be a hormone that is picked up by a G protein coupled receptor. And we talked about G protein coupled receptors when we talked about signaling. So epinephrine, here's your epinephrine in red. It's going to attach on to the G protein coupled receptor, which is going to activate the G protein, which will then activate the adenylyl cyclase, which causes the ATP to be converted into CAMP, a second messenger, which activates the protein kinase A, which leads to the response within the cell. And for epinephrine, the response in the cell could be inhibition of the glycogen synthesis. It could be promotion of glycogen breakdown. So there's different effects that the epinephrine will have, but that's going to be an example of a G protein coupled receptor signaling molecule. Epinephrine is going to have multiple effects. So oftentimes hormones will have different effects, and it could be that you've got different receptors for the hormone on different cells, or you could have different signaling transduction pathways within the cells. So there's going to be lots of different effects. If we look at that epinephrine, that fight or flight hormone, we've got different effects. So if you are this zebra trying to run away from that lion, what's going to happen is in that zebra, it's going to secrete that epinephrine, that fight or flight hormone, and in different tissues, it's going to have a different response. So if you look at the liver cells, that is going to pick up the epinephrine, and it's going to cause the glycogen deposits within the liver to get broken down into glucose, and it's going to be released from the cells. The liver cell is going to secrete or release the glucose into the bloodstream. And so that's going to free up energy for that organism to use right away. So the liver is going to release glucose. And then in different parts of the body, like your skeleton muscles, your skeletal muscle is going to need lots of energy to run away from that lion. And so what's going to happen is the epinephrine in the skeletal muscles is going to cause the blood vessels to dilate. And when the blood vessels dilate, it brings more blood to the skeletal muscles. More blood means more energy, so glucose energy. It's also going to mean more oxygen, so it can do more cellular respiration, and the muscles have the energy necessary to run away from that lion. So your skeleton muscle is using the energy. It's stimulating the ability for the zebra to use the muscles more. But in other parts of the body, you're not going to necessarily need some of those other activities. So, for example, in your intestines, you're not going to be worrying so much about digesting your food as you're running away from the lion. So, your intestinal blood vessels are actually going to get constricted as a result of epinephrine, because you don't need to send nutrients and oxygen to your intestines. You really need those diverted to your skeletal muscles. So epinephrine, while it's dilating the blood vessels to your skeletal muscles, it is constricting the blood vessels that are going to your intestinal area. So because you don't need to do that, right, while you are running away from your lion. So the epinephrine is going to have different effects on different systems and different cells within the body. Okay, when we're looking at hormones, oftentimes, as I said, they are working at maintaining homeostasis. So our body needs to continuously maintain that steady state. So we need that internal balance, regardless of what's happening in the environment. So things like body temperature, blood pH, glucose concentration, water levels, all of those things we need to keep in constant steady state. And that's what homeostasis is all about, is maintaining that internal steady state. And so if we look at things like temperature, we need to be able to maintain our body temperature. Some organisms cannot do it on their own internally, and so they rely on their environment. Some organisms, like humans, can internally regulate your body temperature, and so we're using energy to do that. And that's kind of where we're looking at thermoregulation. So thermoregulation is the mechanism in which we are maintaining our body temperature. So we need to have a body temperature within a certain range. And that's what thermoregulation is all about. Some organisms are endothermic, and they can maintain their constant body temperature regardless of their environment. And so humans would be endothermic organisms. We can maintain our body temperature internally. Some organisms can't do that internally, so they would be ectotherms, and so they're going to rely on their environment. So like that lizard, the lizard, if it is cold, is going to go bask in the sun and lay on that nice warm rock. But if it gets too hot, it's going to go find a shady spot to bring its temperature back down. And so the ectotherms rely on their environment to maintain their body temperature. But all organisms need to keep that steady temperature, body temperature. And so we've got endotherms and exotherms. Used to be called warm-blooded and cold-blooded, but now we kind of go more with the scientific terms, endotherms and ectotherms. So when we look at humans and how we maintain our internal body temperature, if we get too hot, then what's going to happen? So if our temperature goes up, we've got an internal thermostat in our hypothalamus in our brain, and that's going to cause us to sweat. And so sweat is going to use evaporative cooling to bring our temperature back down. Not only do we sweat, we're also going to be dilating our blood vessels that go to the skin so that we can lose heat through the surface of our skin. So that's kind of why our hands swell up in the summertime when we're really hot, because our blood vessels have dilated to our extremities. And that hopefully brings our body temperature back down and into that normal range. If our body temperature drops, then again, we're picking up the sensors in our brain is picking that up, that information up, and it's going to then cause us to shiver. So when we start shivering, the muscles are contracting, they're generating heat, and not only are we shivering to bring our temperature back up, we are also constricting our blood vessels to our skin, and so our skin usually starts to feel cold. Also, it's going to constrict blood vessels to our fingers and toes, our extremities, to keep our core temperature in that range. And so hopefully all of that brings our body temperature up. So in the wintertime, when it's really cold, you might notice that your fingers and toes get cold first because our body is constricting the blood vessels to our extremities to keep our internal core body temperature within that homeostatic balance. So hopefully all of that keeps our body temperature within a certain range. If it's too high, we will sweat. If it's too cold, we will shiver, and it hopefully keeps us in homeostatic temperature balance. So when we have a fever, really what's happening is our hypothalamus is kind of out of whack. And so our biological thermostat set point has been changed, and that's kind of throwing off our internal temperature. And so we're kind of out of balance, out of whack. So when we look at how we maintain homeostasis, usually we're looking mostly at negative feedback, but we've got both negative feedback and positive feedback, and they're going to be working in a little bit different ways to maintain that homeostasis. But with negative feedback, what you've got is a loop that's going to cause the opposite thing to happen. So the response is going to be a way of negating something that has been picked up in your body. So you've registered. So like with the temperature, if your body gets too hot, we do something to bring it back down. And so that's going to be negative feedback. So the opposite thing is happening. With positive feedback, there's a reinforcement of the stimulus. So it produces an even greater response. So you're amplifying the response with positive feedback. And so we'll take a look at some examples of each of those. So here's an example of negative feedback. So what we've got is our digestive system here. And so when food moves into the small intestines from the stomach, we are picking up the fact that that's low pH. So acidic chemicals, we don't really want that in our small intestines. So it's picking up the signal that there's a low pH in our small intestines. And so a hormone gets secreted into the bloodstream, which will then get picked up by the pancreas. So the pancreas is picking up that signal, and it's going to secrete bicarbonate into the small intestines, which is then going to bring the pH up. So a low pH is causing a response that brings the pH back up. And so that's negative feedback. And so low pH causes a response which brings the pH back up. So that's negative feedback because you're causing the opposite thing to happen. When we look at positive feedback, so that's negative feedback with the opposite thing happening. Positive feedback, we're getting an amplification of the signal. And so in this example, we are looking at an example of positive feedback when a mother is nursing her child. So what's happening here is the baby is suckling on the breast, and that is picking up a signal. So it's registering, a sensory neuron is registering the suckling, which is then releasing a hormone into the bloodstream, which is causing the breast tissue to release the milk, which is then causing the baby to suckle even more. More suckling means more hormone, means more breast milk is being released. More milk being released causes the baby to suckle even more, which causes more milk to be released, and more milk and more suckling, more milk and more suckling. And so it's causing an amplification of that process. So more and more milk is being released. until the baby's full and stops suckling, and then it shuts down that process. So that is an example of positive feedback because you're getting an amplification of that signal. Okay, when we look at that homeostatic balance again with the endocrine system, we've got a whole bunch of hormones involved in the endocrine system. We're going to take a look at two hormones that are antagonistic. So insulin and glucagon are antagonistic hormones. What that means is that they work in the opposite way. They're doing the opposite things. So they're going to work together to keep the blood sugar levels in the blood in a certain range, so that homeostatic balance of your glucose levels. The pancreas is going to be involved in secreting both insulin and glucagon, and that's going to help to maintain our blood sugar levels within a certain range. So if we look at this diagram here in the middle, you can see that our blood sugar levels need to be somewhere around 100. And if our blood sugar levels get too high, so let's say we just ate lunch and our blood sugar levels are going up, our pancreas is going to detect that, and it's going to release insulin into the bloodstream. And insulin is then going to cause in our cells in our body, it's going to cause them to take in more glucose from the blood. So more glucose in, they can use that for cellular respiration and get energy for the cell. It's also going to, in the liver, cause the liver cells to take up the glucose and store that as glycogen in the liver. So glycogen is kind of a storage, energy storage, temporary energy storage in the liver. So it's going to take in more glucose, gets it out of the bloodstream, and then all of that brings our blood glucose levels back down into that homeostatic balance, so within that normal range. If, let's say, we've skipped lunch, we skipped breakfast, we haven't eaten anything since yesterday, that's going to cause our blood sugar levels to drop. And so when our blood sugar levels drop, that's going to cause, again, the pancreas to register that we have skipped a meal and we're hungry, and our blood sugar levels have dropped, and that is going to cause the pancreas to release the glucagon hormone, which is then going to cause in the liver to break down that glycogen storage molecule, and it's going to release glucose into the bloodstream. So glycogen will get converted into glucose, it releases it into the bloodstream, and it brings our blood sugar levels back up. So that is a feedback mechanism in which we've got two hormones that are working antagonistically to maintain our blood sugar levels. So negative feedback, antagonistic hormones working together. And so this is a really good example of how hormones work to maintain homeostatic balance when we're looking at negative feedback mechanisms. So if we look again at insulin and glucagon, what's happening is the insulin is reducing the blood glucose levels by promoting the cellular uptake of glucose. It's slowing the glycogen breakdown in the liver, promoting fat storage, not breakdown, and so all of that is reducing our blood glucose levels. The glucagon, the antagonistic hormone for insulin, is going to increase the blood glucose levels by stimulating the conversion of the glycogen to glucose in the liver and stimulating the breakdown of fat and protein into glucose. So again, both of them working together, negative feedback in order to maintain homeostasis. Okay, so that's a quick summary of feedback mechanisms, both positive and negative, and then how hormones and the endocrine system are examples of cell signaling. So with the hormones, we're going to take a quick look at plant hormones and plant responses as examples of signal transduction pathways. You might remember Carlos Linnaeus from ninth grade bio, and with Carlos Linnaeus, he's the guy who came up with binomial nomenclature, but he also was into plants, and what he discovered is that flowers will actually open at different times of the day. And so he put together a flower clock. So you can look at the different flowers and you can look at the clock and you can tell what time of day it is based on whether the flowers are open or not. So it's kind of really cool that we can see that flowers actually do respond to the environment and do respond to stimuli. So if we look at the greening of plants, that is an example of a signal transduction pathway. So when we're looking at plants in the dark, plants will actually be kind of a pale whitish color. They're not really going to be green until they're exposed to light. So when they're grown in the dark, that is called etylation. And then after they have been exposed to light, they would then go through de-etylation. So if you were to leave a potato plant in the refrigerator, it's going to kind of look like this first dark picture over here where it's very pale. So it's going to start to grow in your refrigerator, but it's not going to green up until it's actually been exposed to light, which is kind of pretty cool. So the plants, the potato will grow in your refrigerator if you leave it there for a very long period of time, but it's not going to green up unless it is exposed to light. So when we look at that, it is going to be a signal transduction pathway, but the receptor is picking up light receptors. So we've got phytochrome receptors able to detect light, and then that's going to be your receptor molecule. So the protein is going to be a little bit different than your typical receptor protein. And then we've got second messengers that are going to transfer and amplify the signal from the receptors to the proteins, and eventually it's going to lead to a response. So we can see second messengers, we can see the phytochrome receptors as the first messenger, or actually the light is going to be the first messenger, and then the phytochrome is going to be the receptor molecule picking up that signal, the light signal. So if we look at this diagram here, this is showing you the signal transduction pathway of that de-etylation process. So you can see here the light over here is hitting the phytochrome and it's getting that signal, which is then activating the second messenger, cGMP, which is then activating protein kinase 1, which is then causing a transcription factor to activate transcription, which is going to produce proteins that are involved in the de-etylation process. So the greening up process is going to produce those proteins. If we go back to the beginning with the light, the phytochrome receptors also going to trigger calcium ion channels to open, which will activate protein kinase 2, which is activating other transcription factors, which is also going to be producing the greening up proteins involved in the de-etylation process. So a nice example of a signal transduction pathway in plants. So the response is going to be that signal transduction pathway producing all of those enzymes necessary in order to produce the proteins necessary for the de-etylation process. So it's going to activate enzymes that are functioning in photosynthesis directly. They're supplying the chemical precursors for the chlorophyll production. They're affecting the levels of the plant hormones that regulate growth. So those are the proteins that are being produced as a result of the de-etylation process. Also, looking at plants, there are lots of other plant hormones also that are involved in all kinds of aspects of plant development and growth. When we look at plants, plants will respond to stimuli, and when they do, that's called trophism. When they're responding to light energy, that is called phototrophism. We've also got all kinds of other trophisms like geotrophisms. So they will respond to gravity. So plants will grow roots down and stems up as a result of gravity. When we're looking at phototrophism, we're looking at the plant's response to light. Plants will grow towards the light, and that is phototrophism. Darwin and his son were kind of experimenting and wanting to learn a little bit more about phototrophism. So they noticed this in plants and they did some experiments. And so what they noticed was that when light is shining on the tips of the grass, the plants bend towards the light. And so they did some experiments where they were looking at different coverings of the plant. So first of all, they cut the tip off a plant. And when they did that and shined the light on it, what they noticed was it no longer bends towards the light if you cut the tip off. So then they put a cap on top of it that's opaque and light cannot penetrate it. And they notice it also does not bend towards the light. But if you put a transparent tip on it, it does bend towards the light. So the light can penetrate and it still bends towards the light. And then what they notice is that they put a shield over the base of it, the part that is doing the bending, it still bends towards the light. So that kind of led them to believe that there's something in the tip that's causing that bending towards the light, or the elongation of that region that's bending towards the light. So some other scientists in 1913 did some more continuation experiments based on what Darwin had done. And so what Peter Boyson Jensen did was they cut the tip off it, and they, in some of them, put a permeable barrier that allowed chemicals from the tip to get through towards the base. And if you did that, it does still bend towards the light. But if you cut the tip off and you put an impenetrable barrier between the tip and the base, it does not bend towards the light. So that kind of led them to believe that there's some kind of molecule, chemical substance that's getting from the tip to the stem that's causing it to bend towards the light. So then in 1926, another scientist did some more experimenting with the same process. And what he did was he cut the tip off of it and he put the tip onto an agar cube and let that absorb the chemical. And so when he then put that onto various different cut off tipped plants in the control, obviously it's not going to bend towards the light. If you don't put any of the chemical on the tip with the agar cube, it does not bend towards the light. But what's curious is when he did put the agar cube directly on top of the plant, it also did not bend towards the light. It was only when he put it on half of the plant did it start to bend towards the light. And what he noticed was that the side that he did put the chemical on was the opposite of the side that's kind of bending towards the light. So and this one is bending in the opposite direction. So if you put it on the other side, it bends in the other way. So it's causing, the chemical is actually causing the growth or the elongation of the side that it's on. So when it's bending towards the light, the side that's opposite the light is secreting hormone that is causing it to elongate or grow on this side more than the other side. And what we now know today is that's a hormone called auxin, and that auxin is causing the elongation of the cells on the side that the auxin is on, which is going to be the opposite of the side that the light is coming from. So it will bend towards the light by growing the cells or elongating the cells on the opposite side that the light is coming in on. So plants have a whole bunch of different hormones, and they're all involved in lots of different things. So it could be elongation of the cells. It could be cell division. Some of them are promoting stem elongation, sex hormones, response to drought, fruit ripening, all kinds of things. So ethylene is a very common one that we see examples of, and that's involved in fruit ripening. So if you had, if you bought some green bananas and you wanted them to ripen faster, you could put them in a brown paper bag, and that's going to trap the ethylene hormone in the bag, and then that's going to cause that banana to ripen faster. And so all of these plant hormones are involved in different stages of growth and development of the plants. So plants also have hormones just like humans and other organisms do. And then here's another example of a cell communication in response to stimuli. So this plant is getting eaten by the caterpillar. So it's being wounded. And so what that is doing is that is causing a signal transduction pathway that is causing the plant to release chemicals that are being released into the environment, and that's causing these parasitic wasps to come along, and they're going to kind of attack the caterpillar and lay their eggs in the caterpillar, which kills the caterpillar, which prevents it from eating more of the plant. So kind of a defense mechanism that those plants have evolved to ward off herbivores that are trying to eat those plants. So another example of a signal transduction pathway in plants. So the endocrine system and plants are examples of those signal transduction pathways. And again, with signal transduction pathways, you've got those 3 steps: reception, transduction, and response. So that signaling molecule is being picked up. It's causing a series of reactions or steps within the cell, which is leading to a specific cellular response. Hope that was helpful, and next time we'll talk more about other examples of signal transduction pathways.